will continue to the next presentation. And the presentation is by Kevin Maebe from the Ghent University. And it's on Bumblebee genetics. Kevin, can you please share your screen? Good morning.
Good morning, everyone. I think you can see my screen.
Yes, it's visible. Yes,
it's visible. Okay, perfect. So, good morning, everyone. I am Kevin Maebe of the Lab Agrosology of the Ghent University in Belgium, and I will give a talk about our recent bumblebee genetic research performed in cooperation with the Estonian University of Life Science. And first, here in this first slide, I give a short overview what to expect in this talk. First, I will briefly describe the possible roles of genetic factors in bee decline. Then 2 case studies, the first on temporal stability of genetic diversity, and the second study on the possible correlation of genetic diversity with species evidence and measurement of population structuring. And I will end with some conclusions and a take-home message. So to start, and yeah, I think you all already know by now, worldwide, there are several bumblebee species showing negative population trends, of which several drivers have been identified, such as the agriculture intensification with the loss of food resources and nest possibilities, but also pesticide use, climate change, the new pathogens, and all of these can have individual, but also a combined effect on bee populations and even pollinator populations in general. But okay, also genetic factors can play a role in this bee decline, and populations can become smaller when these previously mentioned drivers have their impact, lowering the genetic diversity within such populations. And due to random factors, such as genetic drift, rare alleles will be lost, reducing the genetic diversity levels even more. Species with such a low genetic variability will have, on the longer term, this theoretically less chance to be able to react and survive future environmental changes. Furthermore, on the shorter term, such populations with low genetic variability will show higher chance of inbreeding. And indeed, the chance of inbreeding will increase and can lead to a decreased fitness due to inbreeding depression. Inbreeding depression is a term of all negative effects of inbreeding. But however, in general, in bees, the most negative effects of inbreeding should be purged from the population in the haploid males. However, a clear inbreeding effect in bumblebees and in bees in general is a protection of diploid males. And therefore, I have to explain a little bit more the haplodiploid system of bumblebees. So, as you probably know, bumblebees have a single complementary sex-determined locus. Males are hemizygote, and females are heterozygote for this loci. When you have homozygotes for this loci, as you can see here, for instance, then you have the diploid males. Yeah. Normally, in a population, there are a high number of alleles for this sex locus, as seen here in this graph. And then the formation of homozygotes is very low. However, when the population decreases, also the number of alleles for this sex locus decreases, and the chance of having homozygotes for this locus and this diploid will increase largely when there is less big populations. What are the negative effects of this diploid males? Well, the effect can be very large. In fact, they can produce sterile offspring. By mating with fertile queens that only mate once, these screens will be lost and they cannot contribute to the next generation. But more importantly, 50% of the diploid eggs that normally are left late in the colony development will develop instead of workers in the diploid males, and this will not contribute to the tasks of workers, so they will not help in the colony buildup. And these negative effects of diploid males can be seen in these two graphs here, in which you can see the outbred colonies, you can see inbred colonies without the formation of diploid males, and you have colonies with the formation of diploid males. And here can be seen that there is a high fitness cost, so the number of offspring is much lower in these diploid producing colonies, and also the mean field survival of these colonies is much lower. So small populations which show chance of inbreeding will have a high number of homozygotes for different loci, will further lowering the genetic diversity, production of diploid males, and these populations come in a so-called extinction vortex. Normally, gene flow can alter the present levels of genetic diversity. On one hand, gene flow between populations or genetic sharing of genetic material between populations. Gene flow can be limited, for instance, by a geographic barrier, as depicted here. When this happens, that leads to a significant substructuring and isolation of a certain normally well-connected population. On the other hand, when there is high dispersal, high connectivity between populations, then this will increase the genetic diversity in a population and will lead to weak population substructuring. So these small populations that will be isolated due to the lower level of gene flow will have a decrease in genetic diversity due to the loss of this buffering effect of gene flow. They will have lower genetic diversity, higher impact of the genetic drift, again inbreeding, and this negative effects will be even more present. So in the previous slide, I showed that there is a low genetic diversity in populations, and this is negative, and this is even more shown in studies with comparing genetic diversity levels and the presence of the prevalences of parasites, as can see here. On the x-axis, you can see the heterozygosity of the host species. Here it is Bombus muscorum on the two graphs on the x-axis, and on the y-axis you can see the parasite prevalence of Locusticaris buschleri on the top graph and Critidia bombi on the bottom, and you can clearly see that parasite prevalence was lower with increasing heterozygosity. Yeah, so these results suggest that when populations lose heterozygosity, the impact of parasitism will increase, pushing threatened populations even closer to extinction. And the same link shown by these studies is also shown in other studies. Here, for instance, in the Cameron et al. For Sidney Cameron, you can see that the genetic diversity here at the right side, the genetic diversity of Bombus occidentalis and Bombus pennsylvanicus is lower than the other species, and these two have indeed also a higher prevalence of Nosema bombi. So again, showing the link between high pathogen prevalence and reduced genetic diversity. So in general, knowing the genetic diversity level within a bee population is crucial to know the health of the population and to make adequate conservation measures. Okay, so this was a little bit more an introduction. There's also that genetic diversity levels within different species are or the stable species that are more commonly present, show higher genetic diversity levels than declining species, populations of declining species showed here. And the hypothesis was made that this lower genetic diversity in the declining species was due to a reduction of genetic diversity caused by the general drivers of bee decline, which certainly in Belgium started around the 1950s due to the agricultural intensification. So there's a drop over time of genetic diversity in these declining species. However, this hypothesis was only based on recent measurements in these bee populations. However, there was an additional hypothesis based on only the measurement of historical bee populations, showing that these declining species already had lower genetic diversity in historical populations, which is the difference was also available there, or visible there. So this led to the hypothesis saying that, okay, populations of declining species have already or always had this kind of lower genetic diversity than these stable species. So in the first study, we investigated this hypothesis, and which of these hypotheses are actually true. And we did this by determining the genetic diversity levels in recent versus historical populations of 4 declining and 4 stable bumblebees in Belgium. Specimens of 5 different locations were genotyped with 16 microsatellite loci, and we compared the genetic diversity from historical specimens around 1913, 1915 versus with recent populations of 2013, 2015. So almost exactly even 1 century later in time. And these are the results. And surprisingly, and in general, our results showed temporal stability of genetic diversity for both genetic diversity measurements. And what are these 2 measurements? Well, these are the most important genetic diversity parameters. So I think I mentioned it, the expected heterozygosity, or the expected mean number of heterozygotes per loci in a population, and the allelic richness, which is the mean number of alleles per loci in a population, corrected for population size. And here in both graphs, you can see on the x-axis the different 8 bumblebee species. And on the y-axis, you can see both parameters, heterozygosity and allelic richness. And the 2 series, you can see the time periods. So you can see here that there is almost no drop or loss of genetic diversity over 100 years for these within each of these species for both parameters, showing so that the genetic diversity are fairly stable over time. But more important, you can see that both the historical and the recent populations of these declining species showed a significant lower diversity here in red than the populations of co-occurring stable species here in the green. This confirms the studies with only recent specimens, but also that with this major difference, in fact, that here we showed that the difference in genetic diversity between both groups was already present 100 years ago. And that this is not due to a drop of genetic diversity due to the drivers of bee decline since the 1950s. So this was our first result, but we were thinking, what could then have caused this observed pattern in genetic diversity? And we thought, as most stable species have a widespread occurrence, and declining species are more restricted, less abundant species, maybe species abundance impacts genetic diversity, with the idea or the assumption that how more specimens are present in a certain population, how higher the genetic diversity levels would be. So in the second study, the aim was to compare the genetic diversity levels of several bumblebee species and to check if the correlation was there with abundance. So to do so, we determined the genetic diversity levels within several Belgian and Estonian populations of 9 bumblebee species. 25 specimens were again genotyped with 16 microsatellite loci. And from each location, we determined also the species abundance. Here we chose Estonia as a second country next to Belgium, because one, the same bumblebee species occur there, but in different abundance than the Belgium. And secondly, because in Belgium, declining species were in Estonia more abundantly present, or even showing increasing population trends. First saying that the abundance of these the 9 species were calculated as the proportion of their abundance versus the total abundance of all bumblebees at that particular location. And here you can see then the relative species abundance of the different bees for each of these locations, with high differences in abundances between these locations. As for instance, you can see here for Bombus pascorum in Moorsel in Belgium, around 60-61% of abundance at that location, and this species is only 10 to 12% present in Estonian populations of Polvama and Harjuma. We also then measured the expected heterozygosity and allelic richness as genetic diversity parameters for these locations, and then to detect the species abundance, and if species abundance influenced and observed genetic diversity levels, we performed linear mixed models in which species, local abundance and location were set as fixed factors and microsat loci as random factor. Okay, but important is that the best model selected on the 18th information criteria showed that abundance was not present in the best model, and this for expected heterozygosity and for the allelic richness. And furthermore, when we compared models with both species and abundance as fixed factors to the models without abundance as a fixed factor, we saw that adding this abundance factor had no impact on our best model. And next to this, we also performed a linear regression of the genetic diversity parameters here in the x-axis, and the richness expected heterozygosity, and in the y-axis, the relative abundance of the bumblebee species. And we found again here only low and non-significant correlation between local abundance and genetic diversity. So this was about genetic diversity and the genetic diversity pattern, and if abundance could explain this pattern. But when we were doing this research, we were also thinking, okay, can we measure the population structure or population structuring within these Belgian, Estonian locations? We did this with 2 programs. The first program is a structured program. It's one of the standards for population structuring measurement. And you can see this here at the left side. And we did also principal component analysis, principal coordinate analysis here at the right for each of the stable species and of the declining species. What can you see here on this structure analysis data? You can see here each specimen is represented by a single bar, and those that belong to the same original population, so for instance, Harjuma, the Estonian population, are grouped within this black vertical line. And this is the Estonian population, is another population with all these individuals. And specimens are assigned to a certain population by the program based on genetic similarities, and are grouped in the same population or cluster by color. So here, the yellow color here shows that all these individuals belong to a separate population. Here, the yellow population. Here, all these individuals belong to the blue population. And what is important here is that you can see that all the Estonian individuals are yellow, belonging to one group, and all the Belgian individuals are belonging, or almost all belonging, to the blue, another group. And this is for Bombus lapidaris. You can see this also for Bombus phosphorum, and you can see this also, this pattern within the principal coordinate analysis, seeing that here the Belgian, here in blue specimens, are grouped, and here the Estonian, here in red, also grouped. So we can see here that both results suggest that there is population structuring on the larger scale between Belgium and Estonia, but not on the more local scale, so between populations of one country, so between Estonian populations or within the Belgian population. And this is for the stable species, which was a little bit surprisingly because a lot of other studies showed no population structuring on the continental scale, but here we detect even in the stable species. And when we go to the declining species, because we did also the declining species, we could see that the structure analysis here right and the PCA showed no or only low population structure. So there is no clear dividing of the Belgian or the Estonian in one color group, so another population. So there is a genetic similarity in each of these populations, which was rather unexpected, as they are not abundant declining species. Why this result occurred here, and it could be that one, the low level of genetic diversity levels observed within these populations of these species might impair the ability to detect this population structuring. Or secondly, it may be an artifact of the limited number of markers used within our study. So we used here 16 microsatellite loci, but performing a study with more loci, for instance, by rod sequencing, where you have several thousand of loci, would greatly enlarge the power of this kind of analysis. So to conclude, we found a temporal stability of genetic diversity in bumblebees during the last 100 years, but confirmed that this high genetic diversity in the stable versus declining species was present. As possible reason, we could discard species evidence as an explanation. We found that species with a low genetic diversity can still reach high species evidence. And about population structuring, we found no population structuring in declining species, but we found I found population structuring within the more stable species on the larger continental scale. So as a take-home message and message for future conservation plans even, our data shows that increasing genetic diversity levels in populations of declining species will be very difficult to do, or to obtain. Furthermore, species evidence is not a good predictor of population health, as species which are locally evident do not mean that they are healthy, as they can have a very low genetic diversity level, and still they could need specific conservation measures, but also the way around. That species with a low genetic diversity level do not mean that a species cannot locally flourish, but then all the environmental circumstances must be ideal to do so. Although there is still a high genetic diversity present within populations of the more stable species, changing landscapes due to human, environmental, or climatic changes may further limit the gene flow between populations of these common species even in time, and thus also conservation strategies must be taken to keep the gene flow and connectivity of these even stable species in check. So this was my presentation. So if you have questions, please do not hesitate to ask.
Okay, thank you. Very nice presentation. I think also quite difficult for many of the people attending, but very interesting, of course. There are quite many questions, so I will try to address some of them. Well, one of the questions is how do you collect the samples? Is it destructive or yeah? So how are the molecular samples collected?
The molecular samples, you mean, of the specimens on the different sites. So the specimens itself were collected during transect walks, the same time, the same method. I think there were trapping nets, so I think it was 1 hour, certain location, catching everything we have. And then it is you do a DNA extraction on each of these specimens. You use your microsatellite loci, and then you amplify this fragment, and then you analyze this with different bioinformatic programs to get this result. So this is quickly in general the thing we do.
Yeah, I think the part of the question was: is it also possible to collect the samples without collecting the specimen?
Not killing them, yeah. So for microsatellites, this is possible, yes. So we did, for instance, by cutting a leg of a middle leg, for instance, of a bee. This is enough to do the DNA extraction of. So normally, a middle leg is not killing the bee.
Okay, so okay, thank you. Yeah. So there's a question from André Kramer from the Julius Kuhn Institute for Bee Protection. The question is, could lower mobility in samplers in the past have led to a less diverse subset of the sampled fauna?
Yes, so indeed, the dispersal capacity is indeed very important in this. When I mentioned with the gene flow, right? So how more the bee is capable of dispersing, transporting, let's say, his gene flow from one population to another, how better and how high genetic diversity can be in this kind of population. So how more dispersal capacity, how better you can try to buffer your negative effect of lower genetic diversity. But in fact,
and another question is,
the dispersal capacities of a lot of bees is not known. So there's only a few bees that or species that we have measurements of. So a lot of certainly the declining species, we do not know how good they can disperse in reality. So it's not known. So this is certainly a field that we would try to work further in.
So another question is if this method can also be used to predict which species are likely to start declining.
Well, in fact, it is the species which have a lower genetic diversity. That's shown that the stable species have a high genetic diversity, declining species have a lower. So in fact, if you see populations with species with a low genetic diversity, these are the ones that are most vulnerable for declining.
Okay, and then last question I have is can you imagine cases in which having a low genetic diversity is good for a species?
It's good for a species. I don't know, but for instance, you have the case of Bombus terrestris invasive in New Zealand, in Tasmania, and it took over the whole island just from a genetic bottleneck of a few specimens. So it is not that you have a low genetic diversity, that you cannot be abundantly present and that you can flourish, for instance. So it's still possible, but then I think all the rest of the environmental circumstances must be ideal. So there must be enough food, there must be enough nest possibilities, and so on. So, but once there is a negative driver then going on, impacting the population, having a low genetic diversity populations with low genetic diversity will be the ones that decline first. So this is a little bit the general pattern.
Okay, thank you.